sealed either with petroleum jelly or a piece of silicone tubing and a glass bead as seen here. Draw the semen completely into the capillary tube, remove the tip, then disconnect the semen-filled capillary tube from the syringe. Place a petroleum jelly plug into one end of the tube with a sterilized spatula. No airspace is needed. The tube can be cut to size if necessary. Be sure to allow space for the petrolatum plug at the other end. To place the second plug, reconnect the tube and move the column of semen flush with the other end of the tube. Detach the capillary tube and place the petrolatum plug. The semen is now ready for shipment. Again, take extra care to avoid contamination of the semen. When preparing for insemination, make sure to properly sterilize and prepare the syringe in advance. Attach the tube to the syringe, leaving the petrolatum in place. This acts as a separator. No airspace is needed. Remove the petrolatum plug on the tip end of the tube before attaching the tip. Be sure to sterilize and rinse the tip with saline solution. Next, collect an airspace and some fresh saline. Now you're ready to proceed with the insemination. The optimal condition is to directly release the queens into nucleus colonies. Queens should be introduced as queen cells or virgin queens, and for convenience can be caged. To facilitate acceptance, spray the queen and bees with a fine mist of sugar water. Be sure to place an excluder over the entrance until egg laying begins. Another option, when direct release is not possible, is to use a push-in cage. Place the cage over emerging brood so young bees will emerge to attend the queen. A review of studies from 1946 to the present comparing instrumentally inseminated queens and naturally mated queens clearly demonstrates the competitive performance of instrumentally inseminated queens and the importance of proper care. Comparison studies measured brood and honey production and queen longevity. The results are categorized into 3 groups. Group 1 includes 5 studies showing equal performance of instrumentally inseminated and naturally mated queens. Group 2 includes 6 studies showing higher performance of instrumentally inseminated queens, which is attributed to the ability to control mating. Group 3 includes 1 study showing higher performance of naturally mated queens. A review of factors affecting queen performance conclusively shows that the treatment of queens has a significant influence. The Group 3 findings can be attributed to the different treatment of queens. Instrumentally inseminated queens in Group 1 and 2 were inseminated at an age range of 5 to 12 days and were given semen dosages ranging from 8 to 12 microliters. These queens were introduced into nucleus colonies or package bees with minimal or no banking. In the Group 3 study, instrumentally inseminated queens were inseminated while 2 or 3 weeks old and confined to cages and bank colonies for another 2 or 3 weeks before introduction. Queens were given 2 small semen doses of 2.7 microliters. The queens were caged and shipped at the start of egg laying, then introduced into large colonies or package bees. The low sperm counts and lower rates of production survival of the instrumentally inseminated queens in Group 3 can be attributed to the methodology used. Queens inseminated past their prime receptive mating age store significantly less sperm. Confinement to cages after insemination also reduces the efficiency of sperm storage and often subjects queens to injury by aggressive worker bees. Queens undergo dramatic physiological changes in preparation for egg laying. Many factors influence the rate of these changes and affect performance. Queens that are inseminated at a young age, allow free movement, and are well attended by worker bees mate with more drones and store more sperm. Gaining proficiency in the technique of instrumental insemination requires practice and attention to detail. The first attempts may seem awkward and frustrating. Concentrate your efforts on mastering each step of the procedure. Success and efficiency will develop with experience. To achieve this requires proper insemination techniques, beekeeping skills, and proper pre- and post-insemination care of queens. Instrumentally inseminated queens are capable of heading productive, full-size colonies. For effective evaluation of breeding stock, it is essential to allow these queens to build strong colonies. Instrumental insemination also enables the creation of specific crosses that do not occur naturally, providing significant advantage to research and stock improvement. For example, a single drone can be mated to one or several queens, isolating or amplifying a specific trait. Varying degrees of inbreeding can be created to produce different relationships within a colony. The ability to pull and homogenize semen from hundreds of drones and inseminate a portion of this to one queen enables unique mating system designs and simplifies stock maintenance. Another advantage of instrumental insemination is the ability to store and ship semen. The shipment of semen, rather than live bees, minimizes the risk of spreading pests and diseases. Instrumental insemination is a valuable and essential tool for honeybee research and stock improvement. With the increasing challenge of pests and disease, selective breeding offers the best long-term solution. Sophistication in instrument designs makes the technique easier to learn and highly repeatable. Add to this our increasing knowledge of bee genetics, breeding system designs, and selection methods. Instrumental insemination truly offers an exciting future potential.